Okay, everybody, let's go over chapter 7 together. All right. So chapter 7, we're talking about the lymphatic system. We're going to talk about the structure and the function. We're going to talk about how it interacts with the circulatory system. And we'll talk about where lymph comes from. You've already heard some of this before when we were talking about the circulatory system. I want you to know that the circulatory system puts fluid out into the tissues, and the fluid that it puts out, it came from the blood plasma, and it's going out carrying oxygen, carrying nutrients. That fluid, most of the big proteins are kept in the blood. Almost all the cells are kept in the blood. Some white blood cells can ooze out of a capillary, but most things are kept in the blood. But the fluid with the nutrients, with the oxygen goes out, and that interstitial fluid, it would just build up, build up, build up. Well, that's not true. That fluid goes out of the capillaries, and then when you get further down the capillary, you have so much, the blood gets more and more concentrated as it loses fluid, and eventually that concentrated blood, the fluid wants to come back into the capillary to dissolve it. We call that osmotic pressure. So fluid goes out of the capillary, it contains oxygen, contains nutrients, surrounds the tissues. That's how the cells in the tissues pick up their stuff they need. And then as you get further down the capillary, the stuff is drawn back in through osmotic pressure, meaning the osmosis, the fluid wants to move. The fluid comes in to dilute all the plasma proteins that got left behind, to dilute the big cells, the formed elements that got left behind, that kind of thing. But what comes back in is never 100% of what goes out. You could build up fluid in the tissues. We call that edema when that happens. The job of the lymphatic system is to drain that extra fluid away and eventually get it back into the blood, into the circulatory system. All right, so let's go over this. Lymphatic system has lymphatic vessels, lymphatic organs. Lymph capillaries are little tiny tubes that they absorb this excess interstitial fluid and get it back to the bloodstream. In the small intestines, the lymph that they're pulling in could be full of fats, and that gives it kind of a milky color. And so they call those capillaries lacteals, as if it was lactate, lactation, lactose, milk looking. So literally they're whitish, and so they called them the tubes to move milk. Anyway, the production, maintain, distribution of lymphocytes in the body. You have to understand, lymphocytes are one of the white blood cell types. We talked about that in circulatory. The littlest one, the L there can remind you of little. So we have two types, B lymphocytes and T lymphocytes, and that's great. But the thing is, a lot of lymphocytes get their name from hanging out in lymph nodes. This is why if a doctor is looking to see if you have an infection, they might be feeling to see if your lymph nodes are swollen. Because if they are, if you've got an infection, your lymph nodes might swell up as lots and lots of white blood cells are made to fight. Anyway, and as we pass all of this lymph through the lymph nodes, that gives the white blood cells a chance to fight any pathogens we've got. So let's move on. All right. Lymphatic vessels are carrying a fluid we call lymph. You already know that's fluid that's drained out the interstitial fluids. It's what was tissue fluid. It says begins as blind-ended lymphatic capillaries. What do they mean by blind-ended? Well, it's just like a dead end. Okay. There's probably a picture coming up. Yeah, that's a horrible picture. Jumping ahead. Yeah, I'm not seeing anything that makes me really happy. I'm not going to jump to another one. Just I'll talk with my hands. Not that I don't do that anyway. All right, so the wall. Imagine my hands are the wall of a lymphatic vessel. Okay, those lymph capillaries, they're closed on one end. So say they're closed here. Now here we have a wall. Well, if there's any pressure at all, they open up and fluid flows in. And then if you try to squeeze that vessel, it'll build up pressure and it'll trap those shut. Okay. They're closed at one end because we want to carry that lymph in one direction. We want to carry it back to the circulatory system. So if your muscles contract and squeeze, just like when we talked about veins, if your muscles get shorter and thicker and that squeezes on a lymph capillary, it's closed on one end. The lymph inside can only go one direction. That's away from the closed end. All right. Anyway, so blind-ended, and as the lymph moves away from the tissues towards the circulatory system, the capillaries form vessels, and then the vessels form ducts. And you have the thoracic duct and the right lymphatic duct. The thoracic duct collects the lymph from, well, you can read it right there. The thorax, the left arm, left side of the head and neck, and it dumps into your left subclavian vein. Let me move that down. Your left subclavian vein. Remember, this collarbone is the clavicle, and the subclavian is the big blood vessel that goes underneath it. It's one of the branches off the aorta, so huge vessel. And that's the one that will eventually, when it goes to the armpit, be the axillary, and then here it'll be the brachial, and here the antebrachial. Anyway, so that subclavian is going to be the spot that the thoracic duct, the duct draining the thorax, dumps into when it returns the lymph to the circulatory system. The right lymphatic duct, it goes into the right subclavian vein. It returns lymph from the right arm and right side of the head and neck. So a much smaller area. All right, let's move on. So there's a pretty picture. And here we have our right lymphatic duct emptying in. This is weird. Let me see. Oh, subclavian vein, not subclavian artery. Okay, that makes sense. Because I was like, that looks like we're getting close to vena cava. Yeah. All right. So thoracic duct. Yeah, thoracic duct empties into left subclavian vein. Okay, good. All right, so lymphatic vessels. The bigger lymphatic vessels are a lot like veins. They even have flaps, valves in them, just like veins. This stops the lymph from going backwards. It can only go in one direction. Remember, we're trying to move lymph away from the tissues of the organs and towards the circulatory system, towards those two big ducts. We're going to dump them back into the blood, dump the fluid back into the blood. All right. And it says the movement of lymph is largely dependent on skeletal contraction. Almost entirely. There is some respiratory pumping, but almost all of it. See, the circulatory system, blood's moved by the heart. And then that stuff that we talked about before with the muscles getting shorter and thicker, and it squeezes on the vessel that's in between, the vein in between. That's helpful for the venous system, but it's absolutely essential for the lymphatic system. The heart does not pump the lymphatic system at all. Lymph moves based on your muscles. So if someone has puffy edema in their legs, one thing they're going to do is they're going to give them a drug that'll, a diuretic, they'll make them pee extra fluid out. But another thing that they will do, they will give them they will tell them that they need to walk, because the more you walk, the more your muscles will squeeze the lymphatic vessels, the muscles in your leg, and draw that fluid out of the legs, move it up against gravity towards the circulatory system, where it dumps in those ducts. All right, so there are two types of lymphatic organs. There are primary and secondary. The primary is saying red bone marrow, thymus. Secondary, lymph nodes, spleen, and tonsils. So, do you remember where I said that the lymphocyte was a tiny white blood cell? Look at it compared to a red blood cell. Almost the same, barely any bigger. I don't know if you can see my mouse, but I'm moving it between a red blood cell and a lymphocyte in the picture on the left. Those four pictures, the left one in the bone marrow is showing both, and lymphocytes definitely way smaller. All right, the thymus, this is where some of the lymphocytes grow up, and specifically the ones that we call T cells. They mature in the thymus. That's why we call them T cells. All right. Your spleen and your lymph node. We'll talk more about the cortex and the medulla. But the lymph node has a layer around the outside. We have a capsule, but then we have around the outside where you see these germinal centers. This is the cortex, and the middle where you don't have it is the medulla. The C in cortex should remind you of the outside, okay? It's near the outside of the alphabet, near the start of the alphabet. The M in medulla should remind you of middle. So the cortex is close to the capsule. It's near the outside. The medulla is in the middle. All right, let's move on. Red bone marrow is where you make most of your blood cells. All right. And in kids, most of the bones have red marrow. In adults, most of the bones have yellow marrow, a little bit of red. B cells grow here. So where I talked about the T lymphocytes develop in the thymus, the B lymphocytes develop in the bone. Pretty straightforward. Also, the T lymphocytes in the thymus tend to travel. The T can remind you of the word travel. They tend to travel in the body a lot more. All right. So the thymus is, it says thoracic cavity superior to the heart. So above the heart, it's right there. It's big in kids, and it gets littler as we get older. It makes a hormone called thymosin that causes those T lymphocytes to grow up. And the immature T cells move from the marrow to the thymus. That's where they grow up. They grow up in the thymus. So they're still made in the bone marrow. That's where they're formed. Then they go to the thymus to grow. Now, it happens a lot more when you're younger than when you're older. Just like you think it's easier to learn languages when you're younger, in the same way, we make and mature a whole lot more T cells when we are younger. We have a bigger thymus. That thymus shrinks as you get older. You lose some of that ability. All right. So the spleen, a lot of circulatory function because it filters blood. It's over here, all right, where it says upper left abdominal cavity. Think over here to the left side. If someone has an enlarged spleen, it might push on their stomach. and actually make it hard for them to eat as much food. That that's a problem that like cancer patients with an enlarged spleen might have. Anyway, so that gives you an idea roughly where it is. Connective tissue divides. You have red pulp, you have white pulp. Macrophages in the red pulp filter things out. Okay. Macrophages are grown-up monocytes. You remember circulatory system, types of white blood cells, never let monkeys eat bananas. That monkeys stands for monocytes. The monocytes, which are already the big white blood cells when you form them, they get even bigger. They grow up to these macrophages. Think of a giant Pac-Man, and it eats not just invading bacteria, but if you have cells that have died when there was a fight between the bacteria and other stuff, or died because there was damage at the wound where the bacteria is coming in, macrophages will eat that stuff. And the spleen is like I said, a spot where you do a lot of filtering of the circulatory system. Red blood cells get removed there when they get old and stiff and have trouble moving through capillaries. Well, they get caught in the spleen, and macrophages will eat the old, worn out red blood cells, so you make new ones. Anyway, a spleen has a thin outer capsule. You don't want it to rupture. Saying so it can rupture from trauma. Yeah, you'll bleed a lot if you do, because it's a filter and you run all the blood through there. All right. So lymph nodes are along the lymph vessels. Remember, lymph capillaries drain into lymph vessels. Lymph vessels they pass through these lymph nodes, and the lymph nodes filter the lymph. They have a capsule around it, and remember, capsule, and just next to the capsule, just inside that, we have the cortex. And so the C in cortex reminds you it's by the capsule, and the M in medulla reminds you it's in the middle. Anyway, the capsule on a lymph node actually has pieces that go down in, like extensions that sink in, invaginations, and that's why they're saying divides it into compartments, because you have these little walls that sink in. Anyway, the lymph nodes are full of macrophages, the big eaters we were just talking about, that eat debris and pathogens. So it helps filter out things that shouldn't be there, and also houses lymphocytes, which fight infections, and cancer cells. Again, lymphocytes, another type of white blood cells. We're talking about two types of white blood cells here. Lymph nodes are named for where they are: in the neck, the armpit, the groin. And again, that's what your doctor is feeling for if they think you have a infection. So lymphatic nodules, that's like a lymph node, but without a capsule around it. And you have those, well, I was about to say Peyer's patches, but that's down below. Anyway, you have those in different spots in the body's tissues. Tonsils are here in the pharynx. You're talking, well, actually higher up there, but the in the pharynx we have the tonsils, and same function as a lymph node. They do the same thing. When you have things come in through your nose or your mouth, and they need to be attacked and controlled, they're your macrophages. They're your lymphocytes. They're your white blood cells. Now, the walls of intestines and the appendix, those have Peyer's patches. Those are like tiny lymph nodes that are spread through the whole organ, and those fight infections that come in if you eat something and it has bacteria all over it, and those bacteria are so tough that they survive the stomach acid and everything, they could invade your body wall. Well, what do? I said your body wall, invade your intestinal wall. So what do? You put concentrations of lymphatic tissue with all of their white blood cells in the wall of the intestine, and then you don't have that problem. a bacteria does get out, it'll probably encounter a white blood cell that'll take care of it. All right, hold on a second. I'm going to pause this. Okay, I'm back. Let me make sure this works. Yep, it's working. All right, so now we're going to do a check our progress. I guess I should have paused a moment ago. Let me pause for a few seconds. You can rewind this and see if you can answer all of the questions. If not, rewind, listen to it again, and then we'll continue. Anyway, I'm going to pause right now. All right, let's continue. Let me try that again. Let's continue. There we go. So we're going to talk about immune defenses. And we'll go over the innate ones. We'll talk about the inflammatory response and complements. All right. So immunity is the ability to kill things that don't belong. And cancer cells are your cells. So technically you could say they belong, but we don't want them. So we want to kill those too. And T lymphocytes, by the way, are really good at that. We have cytotoxic T cells, which kill cancer cells. You might say to yourself, well, why do we have cancer then? The answer is because we err on the side of caution. If we're not certain, we don't usually kill the cell. And a lot of immunotherapy, modern immunotherapy, is waking the body up that, hey, that thing, that's not good. Get rid of those too, even though you weren't certain before. Anyway, there are two branches in the immune system, the innate and the adaptive. Basically, what we're dealing with, the innate is anything that you do automatically, regardless of what the thing is that needs to be destroyed. The adaptive is when you are exposed to something and then you respond by fighting. And usually that means by taking some B cells to remember that infection to respond to it later. We have what we call memory B cells that are important for that. All right, let's move on. So innate or non-specific immune defenses. Examples of that would be physical and chemical barriers. We're talking about things like the skin there. The skin is a physical barrier that keeps bacteria and things. Sorry about the barking dog. The skin is a physical barrier that keeps things out of your body. It also is a chemical barrier because it has some chemicals in it that help to keep bacteria down. An example of that, the skin is slightly acidic, or at least we release chemicals in it that are slightly acidic, and they will help to keep some types of bacteria down. The inflammation response, we don't really care what it is, whether it's a bacteria or a pollen, or if your body thinks something's wrong, it will set off all the histamines and try to wash the thing out and get rid of it. That's non-specific because you do it to anything that seems out of place. And the inflammatory response, we've talked about it before. We are going to make the fluids go at higher speed through the circulatory system and leak out more easily. And then when we talk about protective proteins, they're talking about things like immunoglobulins, which is a fancy way of saying things like antibodies. All right. So they don't have any special recognition of the thing that doesn't belong, no memory of it. By the way, what I'm calling the thing that doesn't belong, the book's calling a pathogen. That should remind you that generates a pathology. It causes a sickness to develop. All right, so these innate barriers are the first line of defense against things that make you sick. The skin and the mucous membrane keep things out. Skin especially, it's tough because it has a lot of keratin, and it's just a protein that makes that the outer layer cells are filled with, and it makes it tough. And we have many, many, many layers of skin. So when we, because it's stratified squamous epithelium, so when we have the outer layer of skin slough off, any bacteria, any microbes sitting on the skin, they come off too. Um, the mucous membranes have cilia. They sweep away mucus with entrapped pathogens. An example of that would be inside your trachea, inside your windpipe. You're carrying the air down. Well, that air, it could have bacteria and dust and things that you don't want to get into the lungs. Well, as the air swirls around, coming down the trachea, bounces off the walls, they're covered with mucus. It's like flypaper. The stuff sticks to it. But you don't want to leave that mucus full of bacteria in the windpipe. So you have little cilia on the cells on the wall that go doo, 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 and they pass the mucus up. They coordinate their movements, pass the mucus up to the back of the throat. You swallow it down to the stomach, and acid kills the bacteria. All right. Chemicals. Acidic accretions from the sebaceous glands on the skin will kill any bacteria, not any, kill some of the bacteria that are sitting on the surface. Also, sweat, saliva, and tears have an enzyme called lysozyme, and that will kill, it will cause some bacteria to lyse, which means to break open or burst. All right, so more chemical barriers. The acid in the stomach can inhibit or kill bacteria. I already told you, the respiratory system, we pass things up to swallow into the stomach to kill. Well, that's reasonable. Now it says normal flora, microbes that reside in the mouth and intestine, prevent potential pathogens from taking up residence. Literally, we have good bacteria living in us, in our mouth, that will prevent bad things from taking up residence. And it says chronic use of antibiotics can make one susceptible to infection by killing off the normal flora. Basically, if we kill off the good stuff, they're not there to prevent the bad stuff from taking over. All right, so here's a summary of innate defenses. And you have dendritic cells, which are antigen-presenting cells. They show the macrophage, like, hey, you see this thing? Yeah, you should be eating those. You should be killing those. All right, that's figure 7.3. Let's move on. So the inflammatory defense is going to be the second line of defense against pathogens. It is mainly neutrophils and macrophages. Now, neutrophils are 50 to 70% of the white blood cells in your body, and that the most common type of white blood cell, and they don't care what it is. They go and they try to kill it. And macrophages, the big eaters, of course, they swallow pathogens and try and get rid of them. But the inflammatory response, the skin's going to feel red, or the body part feels red, hot, swollen, painful. All of those are because what you are doing is delivering lots of blood, full of white blood cells, to the tissue. All right. So why is it red? Because you got so much blood. Why is it hot? Because that hot blood is there. Why is it swollen? Because you got so much fluid coming to the tissue, not just the blood that arrives there, but you have the capillaries get more permeable, the vessels get more permeable, and fluid passes across the wall into the interstitial fluid and stays there. And pain, well, if you swell up like that, you're going to be pinching the nerves. Anyway, so chemicals like histamine, released by damaged tissue cells, mast cells, they make the capillaries dilate and get more permeable. Like I said, that's to deliver more fluid, to wash things away. It's also to make it easier for white blood cells to move from the blood into the surrounding tissue so they can find the bacteria and kill it. All right. 4 hallmarks: redness, heat, swelling, pain. Excess blood flow causes the skin to redden. Yeah, you've heard that. The increased temperature growth will increase temperature will inhibit growth of pathogens. Basically, you are trying to denature or in the bacterial proteins. You're making it harder for bacteria to multiply by trying to heat up the body enough that some of the bacterial proteins don't work as well. The increased blood flow is going to bring a lot of white blood cells to the area. Obviously, lots of neutrophils. They're your fighters. Okay. So figure 7.4 shows this happening. You have macrophages eating things, neutrophils eating things, macrophages releasing cytokines. Yeah, pretty straightforward. All right. So inflammatory response. If you have the blood vessels, especially the capillaries, get more permeable, more fluids can get out, more proteins, things like antibodies. And then you have blood clots that can form if more blood goes to the area, it's more likely to get a blood clot. That would happen in a place where you have a rip, a tear, a puncture. And the excess fluid, which is filling up the tissue, causes the tissue to get swollen, and that pushes on nerves, which causes the pain. Again, you've heard that already. All right, the inflammatory response has white blood cells moving out of the bloodstream into the surrounding tissue. We already said neutrophils and macrophages are really common. Neutrophils are first. They show up, they eat debris, dead cells, and bacteria. Usually that's enough to keep an infection in one spot and stop it from spreading. But neutrophils, because we have so many of them, they also die really, really fast, really easily. And if you have huge amounts of dead neutrophils, that's what pus is. That's white blood cells, neutrophils. If they are being overwhelmed, if they're being beaten, they will release chemicals, cytokines, that will attract other types of white blood cells to come and help them fight. An example of that would be the macrophages, which are really good at eating things. It's in their name. It means the big eater. So they'll eat invading pathogens. Now we have proteins that will help us with the fight, and these are complement proteins. They hang out in the plasma, and it says that they can amplify the response. Some of them hooked up to these mast cells and trigger the release of histamine. I think I've told you before, histamine, we think of, oh, well, his nose was too runny, so he took an antihistamine. Yeah, well, histamine is supposed to be making your nose runny, supposed to be making your blood vessels permeable. The idea is to deliver all the fluid with all the white blood cells to the spot. The idea is to make all the mucus you can to wash the thing out of your nose. If the body is not sure what's going on, but it wants it out in a hurry, it'll probably release histamines. Anyway. So mast cells release histamines, and some of the other chemicals, they attract phagocytes. I'm talking about cells that eat things, meaning white blood cells, like the neutrophils, like the macrophages. And some of them will attack the surface of the bacteria and punch holes in it. This is so cool. The complement proteins, when they do that, they go to the bacteria and they embed into the surface, and they're little C-shaped pieces of protein, but you put one here and here and here and here and here, so they face like that, like that, like that, like that, like that. What that leaves is a little channel, a little tube, a hole in the middle, and water rushes through that hole into the bacteria. The bacteria swells, boom, pops. Sorry, didn't mean to scare you. Pops and spills the bacteria guts out, and bacteria dies. It's pretty awesome. I always compare them to the robot sentinels that they called squiddies in the Matrix, and how they would tear a hole in the side of the ship. Yeah, same thing. These complement proteins will tear a hole in the side of a bacteria and let the water rush in and pop the bacteria. All right, and there's a picture of them happening. Remember I said little C-shaped proteins? You can see how that works. Great. All right, so that was 7.5. Let's talk about interferons. Cells that are infected with viruses will send out an early warning system like, hey, I'm infected, get yourself ready, be ready to fight. And those chemicals are called interferons. The chemicals, when they're detected by other cells that are not infected, they will make methods of fighting viruses. Okay, we're at another one of those memory checks, and I'm going to be quiet for a second. You can pause the video, see if you can answer these questions. If not, rewind it and listen to the video again. Okay, let's move on. So we went through the non-specific immunity. We're now doing adaptive immunity. We're doing things where you are exposed to something and then you respond with methods that are designed to attack just the thing that you're fighting. So antigens and antibodies, I want you to remember two words. An antigen is something that can generate an immune response. It can cause the immune system to ramp up and fight. That's an antigen. The antigen is usually something that doesn't belong in your body. I shouldn't say usually, it is something that doesn't belong in your body. Now, the opposite of that is an antibody. And the word body should remind you it is part of your body. It is one of your body's is one of your body's parts that helps fight infections and things. So the antibody is part of your immune system. And the way I usually describe it is think Star Wars. Think when Darth Maul landed on Tatooine and he's looking for Anakin, he sent out these hunter-seeker probes that would look for and find Anakin. And then they would call home. They would call Darth Maul, like, hey, he's over here, come get him. That's an antibody. An antibody is like a probe. It goes out and it looks for things that don't belong, not just anything. It's looking for a specific thing that it can grab onto. Okay, as an antibody has a specific shape and it's grabbing onto specific molecules on the surface of the bad thing, of the potential pathogen. All right, let's move on. So adaptive defenses, it says, come into play when non-specific defenses have failed to prevent an infection. In other words, that bad thing got a foothold in the body and it's still spreading. Or we have the need for protection against cancer. You can make antibodies that'll fight some forms of cancer. In fact, modern immunotherapies, a lot of times they're making what they call monoclonal antibodies. Your book talks about how that is done. But they are engineered artificial antibodies that can be injected into the blood, travel through the body, and grab onto cancerous cells and say, hey, they do what antibodies always do. They grab onto the thing that they think needs killing, and then they attract white blood cells to help fight it. Some immunotherapy is making special antibodies that probably wouldn't normally be present in high enough quantities to fight the cancer, and releasing them into the body, and then the white blood cells like, oh, look at the antibodies, they found the thing, let's go kill that cell, and then your cancer cell is killed. And that method has a lot less side effects than things like chemo or radiation, a lot less traumatic. Anyway, moving on, they're antibodies and they can respond to cancer. Antibodies are looking for antigens, things that can generate an immune response. Like I said, they're things that don't belong in your body. They could be bacteria, virus, mold, parasitic worms, all that jazz. So your adaptive defenses are looking for proteins in the plasma membrane that don't look like they belong. And if that's the case, well, whatever that plasma membrane is around is probably an antigen. So adaptive ones, this is telling what is self and non-self. In other words, if someone gets really sick and they need an organ transplant, and you're going to give them drugs to stop the rejection, what you're going to do is stop most of these adaptive defenses from working as well, because you're trying to tone all that down so it won't reject the organ. People were like, oh, well, they had an organ transplant. What's often not said is, yeah, they're also going to get sick easier the rest of their life, because their immune system is, we dialed it down a little bit. It just doesn't work as well as it used to, because the drugs that they're taking to keep from rejecting the new organ. All right, so how do these adaptive defenses work? We have B cells or T cells. They're both lymphocytes. Remember, the B cells grow up in the bone, T cells grow up in the thymus. They have specific antigen receptors on the surface. Basically, remember I said where an antibody is a certain shape to grab onto a protein, an antigen? Imagine that same thing, but those receptor proteins, it's like an antibody stuck into the membrane of the T or the B cell. All right. And it's important to know that each lymphocyte has only one type of receptor. It's not the Swiss Army knife of fighting things. It's this lymphocyte knows how to fight this thing. And that lymphocyte over there knows how to fight that thing. And this one can't help there, and that one can't help over here. They have one surface receptor on their plasma membrane, and it fits the antigen protein. It recognizes the antigen protein because it fits like a lock and key. All right. There are two types of adaptive immunity: cell-mediated and antibody-mediated. You can see this. The cell-mediated, T cells kill cells that look foreign. Example, that would be the cytotoxic T cells I was talking about. In antibody-mediated or humoral immunity, let's break that down. The humors is an old school way of talking about the body fluids. So we say humoral, we're talking about we put antibodies into the body fluids. B cells produce antibodies. Actually, B cells make plasma cells, which are like antibody factories. Anyway, so they produce antibodies that bind to antigens in body fluids. Okay. So here we have a comparison of the two, happy little flow chart. And cell-mediated targets things that are on the surface of the cells. Antibody-mediated targets the antigens even if they're free and they're floating around. Cell-mediated, you've got cytotoxic T cells, which I've mentioned several times, helper T cells. That's fine. If you're doing antibody mediated, it's going to be B cells are involved. Either one of those is eventually going to lead to the production of memory T and memory B cells. This is why you can get a vaccine. This is why if you've been previously exposed to chickenpox, you probably won't get chickenpox again. Your body can remember it because you still have surviving long-lived white blood cells. They're called memory cells. They're memory T lymphocytes and memory B lymphocytes. All right, so B cell receptor proteins are, not surprisingly, the proteins on the B cells. Let's see if I can move this. Yeah, I can do that. And I can move this up here. That's also annoying. So cell-mediated and antibody-mediated. B cell receptor proteins are on B cells, no surprise. The antigen, the thing that can generate an immune response, the thing that doesn't belong, it binds to the receptor on only one type of B cell. We already said that. That B cell then copies itself, and the group of identical B cells that you make are called clones. That's all good. And when we have clonal expansion, when we're making these clones, this little clone army, haha, sci-fi reference, the cytokines are being given off by helper T cells, and they tell the B cell to make all those clones. Most of them become plasma cells. I already told you those are little antibody factories that make lots of antibodies that bind to the antigens. They don't just bind to them. You may be like, well, what's binding to it? Well, who cares? They bind to it, and then they release chemicals that attract white blood cells. It's like a little tracker gets stuck to a car, and it sends out a signal, oh, that car is over here, and then the police can come. Same thing. Some B cells will grow up, and instead of being plasma cells, they'll become those memory cells I talked about, that the next time you're exposed to this same antigen, they can wake up and generate an immune response. And that second time that you're exposed to something, your immune response is a lot faster because you have those memory cells. The first time it takes a while to ramp up and get ready to fight, but the second time it's super fast. All right, so here we have the B cells with their receptor proteins on the surface. You can see how different receptor proteins have different shapes, and they grab onto different antigens. All right. And some that are exposed, like, oh, I've just been exposed, that antigen's here again. Then you do clonal expansion. You make a bunch of B cells here for fighting, and you make your memory cell over here. It remembers the infection. This one will last a long. Oh, I said it backwards. Okay, you make your B cells here, which will remember the infection, and you make your plasma cells, which are going to make tons of antibodies they release, which will help to fight the infection. All right. And let's move on. Okay, after an infection has been taken care of, the plasma cells are no longer needed, the little antibody factors are no longer needed, so they have apoptosis, pre-programmed cell death. Basically, we're like, you have fought nobly and we don't need you anymore, so go away. And they say yes, and then they explode. They burst, they pop, and they're done. But you still have the memory cells that live on that will remember the infection and be ready to be fighters in the next generation, the next time that you are exposed to the bad thing, the antigen. All right, so antibodies are Y-shaped. They got two long arms. Each arm has a heavy chain and a light chain. And it says right here, they have constant regions located at the trunk of the Y where the sequence is fixed. All they're saying is that it's just the ends. So here we have our Y. These ends are what are unique and have a special shape to grab onto the antigen. All right. So there we have the heavy chain and the light chain. The heavy chain is longer, so it's heavier. The light chain is lighter, no big deal. Here's the antigen that we have to detect, and we detect it by binding to it. This is an antigen binding site. That means is the part of the antibody that grabs onto the antigen. All right, so moving on. The structure of an antibody. There are variable regions that at the tips that we already talked about, those are what grab on. You saw it in the pretty picture. I used this phrase before and I didn't define it. They say it binds in a lock and key manner. I want you to understand that lock and key Just like a lock has a special shape and only a certain key goes into it, in the same way, an antigen and an antibody, there's a specific shape, and the antibody will only grab onto and detect some antigens. All right, so antibodies, the part of your body, part of your defense system, could be a single Y-shaped molecule that's a monomer, or it could be two of those stuck together, then which is called a dimer. If you had 5 of those little Y's stuck together, it would be a pentamer. And those will have 5 of the little Y's. Those can grab onto lots of the antigens and clump them together. Anyway, they will neutral. It says antibodies coat viruses and toxins, completely neutralizing them, removing the threat. Yeah, that's kind of true. An immune complex, antigens combined with antibodies. What I wanted to get to is that where I said it's kind of true, the antibodies, a lot of times when they cover the virus, cover the toxin, they will, we call it opsonization, which is a fancy way of saying to make tasty, and that will attract those big eating, those phagocytic white blood cells, like neutrophils, like macrophages. And they'll come along and eat the thing. All right. So classes of antibodies: Ig, Im, Ia, Id, Ie. Igs are the immunoglobulins, huge amounts in the blood, a little bit in the lymph, a little bit in the interstitial fluid. They bind to pathogens and toxins. They can go across the blood barrier from mommy to baby and give the baby some of the immunity that the mom has. So Igs cross the placenta. IMs are the biggest and the first one that babies make. You've heard people say, oh, well, breastfeeding is good for babies. It will give them immunity. That is true, and those would probably be IgAs. Anyway, this table 7.1, go through it. I would actually pay attention and know it, because we'll probably pull a couple questions from it. The first column is the name, the second one is where they are, and the third one is what they do. So pretty straightforward. The IgM, the pentameters, pentamers, the first thing that's made by a newborn, first to appear when an infection starts, and the first to go away when the infection is done. And these activate the complement system. Remember, the complements are those little C-shaped proteins I was talking about, that make the tube, and water flows into the bacteria, makes the bacteria swell and pop. All right. IgAs could be monomers, could be dimers. Those are found in body fluids that are secreted, given off mucus, tears, breast milk, and they bind to pathogens and stop them from getting into the blood. IgD are the antigen receptors on B cells that are growing up. E are specific for parasitic worms, and can also cause allergic responses. I know that sounds weird, like, what? But there are actually people who think they can treat immune responses by giving low-level, not too horrible parasites to basically give the immune system something to do instead of attacking the body. I'm talking about using parasitic worms as a treatment for Crohn's disease. All right, so let's move on. Monoclonal antibodies, these are, I told you the book we're talking about these. They're identical, made by the same plasma cells. derived from the same B cell. We can make them in the lab. And for instance, the anti-cancer drug, with air quotes, Rituxan, is a monoclonal antibody. And it says used in medical tests and treatments. Totally. This would be one of those immune system therapies. I know someone who had a form of cancer that caused their cells on the surface to produce a protein called CD20. And they were treated with a drug called Rituxan. It wasn't really a drug. It was a whole bunch of monoclonal antibodies made in the lab. And every so often you'd inject this person with antibodies and they would bind to the CD20 protein on the cancer cells and cause the immune system to attack the cancer cells and kill them. Anyway, here's the pretty picture of how to make a monoclonal antibody. You can see that they are fusing together. This cell here, this hybrid cell, it actually contains part of the cancer cell and part of the immune cell. But the goal is that once you build this hybridoma, it can produce huge amounts of the antibodies that you need. All right. So T cells do cell-mediated immunity. You've already heard that. And each T cell is covered with T cell receptors. T cells can't recognize the antigen. You have to have an antigen-presenting cell bring them that antigen. So that could be done by a dendritic cell, by a macrophage. And the antigen-presenting cells, they'll grab a pathogen that they need to take it in that they're trying to get rid of. And then they'll find a T cell and say, hey, I just ate this thing. Here's a piece of it. I just ate this thing that doesn't belong. Go find those and kill them. They present the antigen to the T cell. All right. So antigen presenting cells break that ingested pathogen apart, and a piece of the pathogen is in the major histocompatibility complex, which is a protein on the surface. There are a couple of them. Major histocompatibility complex 2 proteins are called human leukocyte antigens. And you have them on all the cells in your body. There are 3 general groups. Each one has a protein. Yeah, this is all straightforward. You can read this. Each person who's not an identical twin has a unique combination of HLAs, right? Identical twins having the same genes are going to have the same HLAs. All right. So when we do clonal expansion, you remember that was when we were making lots of copies of the cells, many copies of the T cell in this case. We're talking about T cells. When the macrophage, that's one of those antigen presenting cells, when a macrophage takes an antigen and says, here, this thing that I just ate, you should kill it. When it presents that antigen to the T cell, the T cell complex receptor, the T cell receptor will bind to the antigen. That'll activate the T cell. It will clone itself and make lots of copies capable of finding the thing that was given to it. Some T cells become cytotoxic T cells, some become helper T cells. There's the pretty picture. A cytotoxic, you probably guessed, has cell toxins. Cyto means cell, toxic means toxic, and I don't mean Britney Spears. It has vacuoles inside that are full of perforins and enzymes called granzymes. Perforins, the name does exactly what you think. The little granules in there, the little proteins inside, when they hit another cell, they will embed in the membrane and then expand, punching a hole, perforating it. Of course, that's bad for the cell. So a cytotoxic T cell will find a cell that needs to be killed, come up next to it, and boom, that cell's gonna explode because you ripped holes in it. After binding to a virus-infected cell or a tumor cell, it releases perforins, which punch holes in the plasma membrane, forming a pore. Cytotoxic T cells then deliver granulozymes into the pore, causing the cell to undergo apoptosis, pre-programmed cell death. And this is the cell-mediated immunity. All right. And there's the actual photo of it happening. So we talked about cytotoxic T cells. Now we're going to talk about the helper T cells. Helper T cells release cytokines, and that makes all kinds of immune cells work better. It activates and wakes them up. A B cell, remember, like your memory B cells, they can't be activated without a helper T cell doing its job. HIV attacks helper T cells, attacks white blood cells. And they're the white blood cells that are releasing cytokines to wake up an immune response in lots of other white blood cells. So HIV is actually shutting down the thing that generates or stimulates a huge immune response. That's how it shuts down the immune system. It gets rid of the thing that helps all the other cells do their job, sends messages. All right, this is another one of those check your progress things. I will be pausing in a moment. You can read over the slide, see if you can answer all the questions. If so, great. If not, rewind, rewatch again. But I'm going to pause a moment, then we'll continue. Okay, let's move on. Let's talk about the acquired immunity, the difference between active and passive, the importance of cytokines, which we've mentioned in passing already. All right, so you can have natural immunity, like, oh, I got an infection and now I'm immune to that. An example of that would be like chickenpox, or artificially through medical intervention. An example of that would be something like, oh, that person got bitten by a rabid squirrel. Give them injections of antibodies so their body can fight the infection. Anyway, so active and passive. Active immunity would be the example where your body learns to make immune cells. Learns to make antibodies. Passive is when you are given the antibodies, but you don't make them yourself. So in active immunity, you're going to be making antibodies against an antigen. And this can happen when you're immunized. You get a vaccine, and it will either have some of the antigen or from the pathogen, or could have a weakened form of the pathogen itself. We try, if we can, not to use live vaccines like that. If you're using just a piece of the pathogen instead of the whole thing, you're less likely to cause sickness, illness. All right. The first response to an antigen is the primary response. I already told you that's slower. The secondary response, which is based on your memory B cells, memory T cells, maybe getting booster shots, that is a faster response. So you can see the second response is bigger, stronger, faster. All right, moving on. Passive immunity is when you're given antibodies to fight a disease, because it's not because you're not making the antibodies yourself. It's temporary. The book's example of this is breastfeeding. I already told you the IgA, IgB, IgG antibodies that are present in the breast milk. Totally true. But also below that where it says receive antibodies by injection. That's what they're going to do if you're bitten by an animal. They think that it's rabid. They're going to not wait to test and find out because rabies will kill you. They will instead inject you with antibodies that will fight the rabies virus. Okay, cytokines are signaling molecules. They're made by T lymphocytes, macrophages. They regulate whether or not you make white blood cells and how they work. Examples of that would be interferon and interleukin. They're used in all kinds of medical treatments. Oh, here's the check your progress. So again, see if you can answer these questions. If not, rewind and watch again. I'll be quiet for a second. Okay, let's move on. Hypersensitivity. This is not just allergic reactions, this is autoimmune diseases, okay? This can be bad. I'm talking about things like lupus and Crohn's disease and type 1 diabetes, stuff like that. So hypersensitivity is when the immune system overreacts and it attacks the body. Allergies, incompatible blood type, tissue rejection. Remember, that's why I talked about you get an organ transplant. Autoimmune disorders, that's the stuff like the type 1 diabetes. And so allergies are when you're super sensitive to something that normally is harmless. I'm talking about things like pollen, food, animal hair. And you get an immediate response within seconds of being in contact with the antigen. Some of these are irritating, like pet hair. Some of them will kill you dead. I'm talking about stuff like a peanut allergy. All right. They are caused when the IgE antibodies on receptors on mast cells, basophils, they release histamine. I already told you the idea is the body is like, I don't know what it is, but it's not normal. Get rid of it. Wash it out. Wash it out. It's just that your body's overreacting because the thing is pretty normal. All right, so if the pollen is an antigen, the histamine is going to make your mucous membranes run, runny nose, watery eyes, hay fever. You're trying to wash that stuff out. With asthma, you have airways constrict and there's wheezing. Food allergies, nausea, vomiting, diarrhea. Like I said, things like a peanut allergy are super, super bad. All right. Anaphylactic shock is a super strong immediate allergic reaction. If the thing that's the allergen, the thing that generates the allergy, enters the bloodstream, your body is going to try and dilate all the blood vessels at once to let the maximum amount of blood flow through and wash the bad thing away. The only problem with that is you don't have enough blood in your body to open all the blood vessels at once. And so you're going to get this huge drop in blood pressure because you don't have enough fluid for everything to be open at once. And this is caused by the dilation and by the release of lots of histamine, so fluid leaves the blood. You could take epinephrine to counteract the reaction to make you less susceptible to end the anaphylactic shock. That's the best way to put it. You can also have a delayed allergic reaction. That's caused by memory T cells, uh, uh, where you got the exposure. An example like poison ivy. All right. Tissue rejection. If you get a liver transplant, get a heart transplant, they give you the new organ and it's not yours. And the body will recognize that it's not yours. And the cytotoxic T cells will attack it and destroy it. And that's a problem because you just got this new organ. So they can give you drugs, immunosuppressive drugs, that will stop the cytotoxic T cells from attacking that tissue. Now, when they do, they're going to suppress the immune system. So you'll always have to be more careful when you go out in public. Some of these immunosuppressive drugs inhibit cytokines. Tissue rejection, xenotransplant, that'd be an extreme example. That's where you're taking an organ from some other animal, not a human. We don't do that as much as we used to, or so we don't do that much, I'm aware of. Obviously, there's this really huge famous example where Loma Linda University took a baboon heart and transplanted it to a human who needed a heart transplant and they couldn't find one. Now, this thing here where some organs can be grown in a lab, we have that happening in Florida. The folks at Shands up in Gainesville are trying to figure out a way to take a pig kidney, wash away the pig cells, put human stem cells from a patient who needs a kidney on the protein scaffold that's left, and grow a new kidney that has all the cells derived from the patient, and then they could transplant it in and there'd be less tissue rejection. Anyway, moving on. So severe combined immunodeficiency, you have humoral immunity or cell-mediated immunity, either one or both. I should say, both of them are not working properly. They could be less than normal, or they could just be gone. In that case, even a little infection, it can knock you out, kill you. The way they treat that is either a bone marrow transplant or gene therapy. These are the people who, before we had those treatments, would live their lives in a bubble. Okay. Acquired immune deficiencies can be caused by infections, chemical exposure, radiation. A really famous one, of course, would be AIDS, which is caused by HIV, the human immunodeficiency virus. AIDS patients have all kinds of infections and they're at a high risk of cancer. They'll get cancers that most people don't. Like, it's ultra rare to see, I think it's called Kaposi's sarcoma. Anyway, it's a very rare cancer, but it's fairly common in people with HIV because their immune system doesn't fight this cancer and hold it at bay. All right, so immune diseases. We already talked about some of these, but we'll talk more. In autoimmune disorders, cytotoxic T cells or antibodies from the B cells attack the body's cells like they were foreign. Basically, your immune system is hypervigilant, it's wary, it's wanting to attack, and it attacks too well. It's too good at the job, and it's destroying good cells. This could be rheumatic fever. And it says antibodies induced by bacterial infection in the throat also attack the heart muscle. You could have you could have rheumatic arthritis, antibodies against the joints. In other words, your joints are part of your body. They're good. Nothing should mess with them. but for some reason you start developing antibodies that bind to and attack the joints. All right. And as the joints of someone's hand get destroyed, that's what the hand can start to look like. Okay. Systematic lupus, erythematosus. Most people call it lupus. Facial rash, kind of a red butterfly thing going on. Fever, joint pain, but it's not just a skin thing. It can attack the central nervous system, the heart, the kidneys. It can kill you. You're making antibodies that attack DNA, and since almost all the cells in the body have DNA, lupus can show up anywhere in the body. It's bad. Myasthenia gravis, antibodies attack the muscles get weaker, and then you die when the diaphragm muscle stops moving to move air in and out of the lungs. A bad way to go. Multiple sclerosis, the T cells attack the myelin sheath. We're talking about the support cells around the nerve, and it attacks them, it weakens them, and then you get scarring, non-functional, the nice myelin sheath cells, things like Schwann cells, those are replaced with non-functional cells, and then you can't transmit a signal down the nerve so well. This could cause systems to not work, muscular weakness, double vision. It's not good. We're guessing that that's an autoimmune. We don't know. The reason they're guessing is that the drugs that appear to be useful are immune suppressors. All right, here's and by the way, the one they're not mentioning there, type 1 diabetes. The body attacks and destroys the cells in the pancreas that produce insulin. So, all right, I'm going to pause. Will you check to make sure you can answer these, and then we'll move on. Oh, and that was the end. We are done with this chapter. All right, then let's end this, and we can move on to the next one.